By definition, the heme agglutination inhibition test. The heme agglutination inhibition test is a laboratory technique used to measure the presence and the concentration of antibodies against virus that cause heme agglutination, such as influenza virus. The procedures test involve mixing the virus with red blood cells and the serum sample. If antibodies are present, they will prevent the virus from agglutination, clumping the red blood cells. Heme agglutination inhibition test has several important applications. Number one, diagnosis of viral infections, detection of antibodies. The HI test help identify past infection by detecting antibodies against specific virus in patient serum. Epidemiological studies, it's used to monitor the spread of viral infection within population. Vaccine efficacy, and this is the important point in our discussion, how we can measure the vaccine efficacy by measuring the immune response. Immune response measurement. The test measure the immune response to vaccine by quantifying the level of antibodies produced after vaccination. Vaccine development, it aids in evaluating the effectiveness of new vaccine during a clinical trial. In research, virus antibody interaction, researchers use the HI test to study how antibodies interact with virus, which can provide insight into viral mechanism and immune responses strain identification. It helps in identifying and differentiating between various strains of viruses, particularly in influenza research. Quality control, vaccine production. The HI test is used to the quality control of vaccine production to ensure that vaccine include the desired immune response. ELISA, enzyme-linked immunosorbent assay, is a widely used laboratory technique for detecting and quantifying the substance such as protein, antibodies, and the hormone in the biological sample. The purpose, ELISA is used to diagnose infections, measure hormones level, and detect allergen, among other applications. The test involves binding an antigen to a surface, adding a specific antibody linked to an enzyme, and then adding a substrate that the enzyme can convert to a detectable signal, usually a color change. The interpretation of the vaccination result by ELISA. This is usually done by evaluating the three main key components of the immune response after vaccination, which are intensity of the response, mean titer, uniformity of response, coefficient of variation, percent, and the persistence of response, mean titer response over time. How can we interpret the vaccination result? By using ELISA, we can use the mean titer, CV percent, mean titer response over time. Intensity of response, mean titer. Doper develop sufficient titer levels that are in the expected range for the used vaccine. These expected titer following vaccination are often called baseline titer. The baseline titer values may vary according to types of birds, age, vaccine type, vaccination program, and other factors. Therefore, one should make their own baselines for their own vaccination program and local conditions. Uniformity of response, coefficient of variation, percent. Is the vaccine actually getting to the all birds or not? The general guidelines for percentage coefficient of variation, CV percent, following vaccination as follows. Most important surgical, the ELISA, one of the most, as we mentioned, one of the most important serological tests to use to monitor the vaccination response. Persistence of response, mean titer response over time. Do titer persist long enough over time, or is another vaccination needed to boost titer above minimum protective level? This is a very important question, and ELISA can reply this question efficiently. So, percentage of CV and uniformity less than 30%, excellent. From 30 to 50, good. Greater than 50, need to improve. The baseline titer in broiler, Newcastle disease virus, vaccine type, mean titer range, and suspected titer infection. And this is for ND and IBD and IBV. The mean titer range and the weeks after vaccination to test. The rapid plate agglutination test. The rapid plate agglutination test is a diagnostic method used primarily in veterinary medicine to detect antibodies in the serum of animal, particularly poultry. It's used to identify infections such as mycoplasma and salmonella in poultry by detecting specific antibodies. The test involves mixing a serum sample with a suspension of antigen on a plate. If antibodies are present, they will bind to the antigen and cause visible clumping. Application commonly used for monitoring and controlling disease like Mycoplasma gallisepticum, Salmonella ballonarum, gallinarum in poultry flock. Benefit: the RPA test is valued for its speed, simplicity, and cost effectiveness, making it a particular choice for large-scale screening poultry farm. The agar-gel precipitation test. Agar-gel precipitation test, also known as Octarlone test, is a method used to detect the presence of specific antigen or antibody in sample. It's used to identify and to quantify antigen or antibody, often in that context of diagnosing infections or studying immune response. Set up: antigen and antibody sample are placed in wells cut into an agar gel. Both the antigen and antibody diffuse through the gel, and it's used to detect the antigen or antibody.